Welcome to Chapter 12, DNA Replication, Part 2. Applying replication to things like, forensics and sequencing the human genome and basic lab protocols. So it's about polymerase chain reaction. So from the beauty of Yellowstone National Park arose the molecular technique known as polymerase chain reaction. And what we see here is one of those hot pools of liquid. It's at 160 degrees Fahrenheit. That's pretty darn warm for you and me. But for little bacteria called Thermus aquaticus, it's the perfect temperature. And we took those bacteria out of there and we found that they have DNA polymerase that can act at these high temperatures and not being denatured. So we get what is called Taq DNA polymerase from this little guy called T. aquaticus. We take the first three letters, Taq, to name the special DNA polymerase that can work at these high temperatures. It's a thermophilic bacteria, Thermus aquaticus. It can withstand heating without denaturing. The half-life of the polymerase is 40 minutes at 95 degrees Celsius, which is almost boiling, 203 degrees Fahrenheit. And it replicates at a rate of 1 kilobase, which is 1,000 base pairs, in 30 seconds. And that's pretty darn fast. At 72 degrees Celsius, which is 161 approximately degrees Fahrenheit, which coincidentally is the temperature at which this bacteria likes to live. Excuse me, this work was published in 1976. This DNA polymerase has 5' to 3' polymerase function, but it lacks the 3' to 5' exonuclease function that I was quickly going over in the last video lecture. And that's to correct mistakes. So it can't correct mistakes. Therefore, it is considered a low fidelity enzyme. And it makes errors at a rate of 1 in every 10,000 nucleotides, which honestly, that's a pretty low rate of mistakes. But, it does make mistakes. So polymerase chain reaction, or PCR for short, there's an animation. It's on Blackboard, but it's also in your chapter. It's chapter 19, the one video from chapter 19 that's in your Sapling Plus, and you have a you need to watch and study that video and then take the quiz, the activity quiz on it. We can go and look at it in a second, but this is just a little picture showing you've got double-stranded DNA over here on the far left. What happens is you have little tiny pieces of DNA that are only 22 nucleotides long called primers, and we use DNA primers instead of RNA primers, as was we use RNA primers in the cell. And then you synthesize your new DNA using the old DNA template in semi-conservative fashion. Taq DNA polymerase is creating these new DNA strands. Then you denature it, you heat it up, and you anneal the primers again, and then you elongate the DNA. You denature the double-stranded DNA into single-stranded DNA. You anneal or complementary base pair the primer to the denatured DNA. You reduce the temperature and elongate the DNA using Taq DNA polymerase. And so what we have is exponential growth of the number of template DNAs every time we go through a cycle of polymerase chain reaction. Okay, and they still obey the rules of complementary base pairing and anti-parallel interaction. And 3' hydroxyls must be at the end of the DNA oligonucleotide primers in order for the DNA polymerase to start working and making new DNA. So every time we have an exponential relationship, so the after 40 cycles, it's 2 to the n, where n is the number of cycles. So it would be 2 to the 40th power. So 2 to the first power is 2, 2 to the second power is 4, 2 to the third power is 8, and so forth. So after 40 cycles, you'd have, oh boy, 1.1 times 10 to the 12th DNA template. So that's a lot. So exponential growth is shown in this table, where for every cycle, you're going to get 2 to the n numbers of DNA molecules, where n stands for the number of cycles. So if you've been through one cycle, it's 2 to the first power. If you've been through 10 cycles, it's 2 to the 10th power, which means you're going to have over 1,000 DNA molecules that you make during this reaction. So here's the formula for growth, which you should know for the exam, which is 2 to the n, where n is the number of cycles. When we compare DNA replication in a cell to DNA replication in a tube, here's the differences and similarities. So in the cell, you're going to start with a double-stranded DNA template. You're going to have some proteins called initiator, helicase, gyrase, and single-stranded binding protein, just as we saw in the animation. You're going to have an RNA primer that provides a 3' hydroxyl group. You're going to have, in a bacterial cell, DNA polymerase 3 that's going to elongate the DNA. Using deoxynucleotide triphosphates, which we abbreviate DNTPs. So these are the A's, the T's, the G's, and the C's. More enzymes like primase, DNA polymerase 1, and ligase. And how does that compare to DNA replication in a tube, or PCR? Well, you still start with a double-stranded DNA template. You have to use heat instead of all these proteins to break apart the double strands into single strands, denature the DNA. Then you're going to have a primer that's going to complementary base pair to that those single strands of DNA, providing the 3' hydroxyl. Tac DNA polymerase instead of DNA polymerase 3. You're still going to have DNTPs, deoxynucleotide triphosphates, the A's, the C's, the G's, and the G's. And you're going to have magnesium ions. So in your cells, you're going to have magnesium ions too, but you know, you actually have to add it to the tube in this case. It doesn't come with. So magnesium ions, salt buffer, and water. So you have to have the right pH and all that jazz. So, you know, some of the highlights are, instead of all these proteins in the cell, in the tube, we've got heat. Instead of RNA primer in the cell, we've got DNA primer in the tube. Oh, and the last, of course, was DNA polymerase 3 versus Tac DNA polymerase. And DNA polymerase 3 has exonuclease activity, so we're going to have pretty much perfect replication, whereas Tac DNA polymerase does not have exonuclease activity, so you're going to get some mistakes. Job of PCR component is to heat. We're going to raise the temperature and break those hydrogen bonds so the double-stranded DNA becomes single-stranded DNA. When we lower the heat, we're going to allow new hydrogen bonds to form between the DNA primers and the DNA template. And they're going to do this through complementary base pairing. The 3 basic steps in PCR are denature, so that we're going to unwind the DNA with using heat. Double-stranded DNA is going to become single-stranded DNA. We're going to lower the temperature so that we can get complementary base pairing, which we call annealing of the deoxyribonucleotide primer. to the DNA template. And then the third step is elongate the complementary DNA sequence. And we provide optimal heat for the Taq DNA polymerase to do that. So we have to raise the heat slightly from the annealing. So we're going to heat it really hot, cool it down, and then warm it back up a little bit. Typical temperatures include denaturing at 94 or 95 degrees Celsius, which is very close to boiling. Annealing is about half that temperature, between 50 to 65 degrees. This allows the DNA primer to complementary base pair to the DNA template. And then we raise the temperature just slightly because the Taq DNA polymerase likes the temperature 72 degrees Celsius, which is similar to the pool of steaming hot water it comes from. So PCR background, it was invented back in 1983. Dr. Q was alive then. Yes, she was. And it was invented at Cetus Corporation. Transformed DNA science from difficult to possible for most anyone. And if we, well, a lot of times we do that in lab class. Hopefully we'll be doing that in lab class. If not, you'll be doing it in a virtual lab. There are four main areas of impact: gene mapping, engineering DNA, making things like genetically modified organisms, GMOs, DNA sequencing, and gene detection, like forensics, like who done it, right? Who's that blood splatter from? The guys who invented this lovely technique got a Nobel Prize in 1993 in chemistry for the polymerase chain reaction. Carrie Mullis is from North Carolina, and Michael Smith here, Canada. Oh, Canada. Okay, that's all. So summary: replication occurs at high rates and it's accurate, but spontaneous mistakes occur, resulting in genetic variation or mutation if we don't correct them. So sometimes we can't correct them with the exonuclease activity, and they do stay in, and that's how we get genetic variation that can sometimes be a mutation. PCR is DNA replication in a test tube. It's in vitro rather than in vivo, and it's important in science and in law. And now I'm just going to try to go quickly over to your Blackboard site. Well, I'm going to go to an old Blackboard site, honestly, because I don't think I've got it loaded in yours at this point. So hopefully that comes on quickly. Okay, there we go. And go ahead and download this.
A sample of chromosomal DNA, also called genomic DNA, can be used as the starting material for the polymerase chain reaction, or PCR. With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest, or target DNA, is indicated in red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Taq polymerase. The primers are typically about 20 nucleotides long and are complementary in sequence to the ends of the target DNA. Taq polymerase is derived from hot springs bacteria and can tolerate the intense heat of a PCR reaction. A PCR reaction lasts several hours and typically consists of 20 to 35 repeating cycles. A cycle begins by heating the reaction mixture to 95 degrees Celsius. The heat denatures the DNA, breaking the hydrogen bonds that hold the strands together. After denaturing the DNA, the temperature is reduced to around 60 degrees so that the primers can form hydrogen bonds, or anneal, with their complementary sequences in the target DNA. Note that the primers and the target DNA follow base pairing rules. An adenine, A, pairs with a thymine, T, and a cytosine, C, pairs with a guanine, G. In the next phase, the temperature is raised to 72 degrees Celsius. Taq polymerase functions optimally at this temperature and begins polymerization, adding nucleotides to the 3' end of each primer attached to the DNA strand. After one complete cycle, there are 2 double-stranded copies of the target DNA. The PCR reaction mixture contains many copies of the primers and an abundant supply of nucleotides to perform many additional cycles. After a second cycle, there are 4 copies of the target DNA. After cycle 3 is finished, there are 8 copies of the double-stranded target DNA sequence. Note that only 2 of the double-stranded copies consist of just the target fragment. The others also include flanking DNA regions. As the number of cycles increases, the products consist of a greater proportion of fragments with just the target DNA. After 4 cycles, half of the fragments consist of just target DNA, and half of the fragments also contain flanking DNA. With each additional cycle, the number of copies of our target sequence doubles. At the end of cycle 25, there are more than 33 million copies of this double-stranded target region.
Okay, so, and of course you've got that animation in...
a sample of chromosomal DNA also...
Stop sapling, and you have a quiz to take on that. So please do that, and I'll be glad to answer any questions during our next virtual meeting.